So Jian Ma comes to us from Carnegie Mellon University in Pittsburgh in the US. He's a computational scientist. He runs the Computational Genomics Laboratory at Carnegie Mellon. But as I mentioned to you, he's now a key person in this 4D nucleome, as he was awarded together with many others. But he is one of the coordinators of one of the centers, this 4D nucleome. You see some of the slides here. So this 4D nucleome has several centers around the US. And these centers are groups of researchers coming all together, research groups coming all together. And he leads one of such centers in this second phase. And I guess Jian Ma will tell us a bit more. What I find very interesting on Jian Ma's work is that he touches on almost in all topics related to the analysis, computation, visualization, and things that you have to do with all this sort of data. And recently, he also released one of the, what I think, most impressive visualizers of all this data, the nucleome browser, so sort of a three-dimensional genome browser. So I don't want to say much more. I want to give the floor to Jian Ma. He's going to talk for about 30 minutes, and then we will have time for questioning. So feel free to ask anything at the end of his talk. Jian Ma, the floor is yours. Thank you for being here.
Oh, thank you. Thank you for the kind introduction, Mark, and thank you for the invitation and the opportunity to present today in this INC mini symposium. So as Mark mentioned, I will first give you a very brief introduction of the NIH 4D nucleome and also our center project. It's a at NIH, it's a mechanism called UM1. It's a center project that I've been involved in. Actually, Ting Wu, who will be talking shortly, is also part of this center project. And then the second half of my presentation, I'll introduce two recent work from my research group that may be of interest to many of you in the audience. The NIH 4D nucleome project, I think, started over 5 years ago, 2015, and this is funded through a common fund of the NIH, and the aim is to understand the principles underlying nuclear organization in space and time. So 4D, the fourth dimension is the time that we want to look at the changes of the 3D genome over time in normal development or disease progression, and to understand the role of nuclear organization, what their roles in gene expression and cellular function, and how these changes of nuclear organization affect normal development and also various types of diseases. And in second phase, the first phase mostly focused on technology development, method development, but in the second phase, there's a slight shift of the technology development to more sort of the application of these technologies, including experimental and computational, to various types of contexts, including disease models, to look at the variation of 4D nucleome. And so, as I mentioned, the program develops technologies, resources, experimental techniques, computational methods, integrative analysis tools, and including visualization tools, resources, and these platforms that can disseminate these kind of various types of resources and data in multiple modalities to the rest of the community. And this is the front page of the 4D nucleome project at the NIH. For the consortium itself, there's so in the second phase, this started late last year, and then it's going to run until 2025. These are the type of awards or the grants that are being supported in the 4D nucleome phase 2. There's one organization hub that manages the communications within the consortium. Also is responsible for the outreach activities to the rest of the research community. And there's a data coordination center that collects all the data that being generated in the consortium and to compile and organize these data so that people within the consortium, also in the rest of the community, can have easy access to these data and resources for their own work. And so there are other 4 types of funding mechanisms that with different scientific, slightly different scientific focus. One is on real-time chromatin dynamics and function to look at the dynamics, and a lot of these projects are using state-of-the-art imaging technology to study 4D nucleome. And there's also 4D nucleome centers for data integration, modeling, and visualization. And the center that I will briefly introduce to you is part of this for data integration centers. There's also projects that with a specific focus on particular type of diseases, and there are many projects are developing, are applying genomic and also sometimes imaging technology to look at disease models. And in this program in phase 2, there's also quite a few projects awarded to new investigators. Those are the investigators who have never received NIH grants before, but they have a strong track record in developing various types of techniques for studying the nuclear architecture. And for the organization, there's NIH program leaders from several different institutes within the NIH overseeing the development of the consortium. And there's a steering committee organized by investigators in the consortium, together with input from external program consultants, and also the funded research groups that I mentioned from the investigators funded from awards I mentioned before. And I think one unique part of the 4D nucleome is that investigators and research groups within the consortium, they would form these so-called working groups. These are actually organized not based upon the funded projects, but they are formed from the interest from the group. For instance, there are working groups on development. There's a working group on developing computational, like machine learning predictive models from the consortium. So these working groups will come together to meet regularly and foster new collaborations within the consortium. And there's also interest groups. These are also similar to working groups, but working groups there are milestones and deliverables that will report back to the NIH. So this is so I will briefly introduce our center in a minute. And there are a lot of resources within from the 4DM actually are public facing. They're everybody around the world can have access to that. There's a web portal to disseminate information about data protocols. There's resources from the project. Very importantly, there's a data portal that is organized by the Data Coordination Center. So it hosts most of the data, all the data from the 4D nucleome network. And then the center also, this portal also organizes related data sets from other types of projects within the funded by the NIH or from the community, as long as there is some connection with the data being generated from the 4D network. So here's a screenshot of this 4D nucleome data portal, and you can have access to that as well. And there's other resources like internal wiki and directories to organize the consortium activities. There are resources on cell lines and protocols to make sure that people within the consortium, if they're working with the same system, making sure that they're using the same cell lines, and at least they are documenting the metadata within the same conventions. And very importantly, in 4D nucleome, all the research coming supported by the 4D nucleome project will have to be submitted to bioRxiv. At the same time, you submit to a journal. So it really promotes open science and with fast access to the community for the research coming out of this consortium. And there's also a lot of different policies that are discussed within the consortium, and sort of a lot, all of us will follow, including publication policy. So the one thing I mentioned that when you submit a paper supported by 4DN to a journal, you have to also submit to a preprint. That is part of the policy of the 4DN. So I will highlight the projects that I'm involved in, and this is a UM1 center project I mentioned, is a data integration project, and the title is Multi-Scale Analysis for the Nucleon Structure and Function by Comprehensive Multimodal Data Integration. We have 9 investigators around the country, and actually also we have colleague Jason Swellow from Europe, and all these labs have very different expertise, but scientifically, we're also geographically very distributed, but scientifically, actually, we're highly complementary and coherent, and we have all worked together in many different ways in the past. I'll briefly mention what we are doing in this center. So, as you all know, genomic mapping have produced the detailed characterizations of sub-megabase scale chromatin interactions, for instance, TADs and loops within the cell nucleus. And based on CHi-C analysis, we know that large-scale chromosomes are spatially organized into active and inactive A and B compartments largely. But this type of separation is rather binary, and we thought that it's perhaps too overly simplistic. And that's something that this whole project co-center is focused on, is to refine this A and B definition to more fine-scale characterization of how the chromosomes are organized spatially within the nucleus. And I don't have to provide too much background information to this particular audience. We all know that in the nucleus, in addition to chromatin, there are many other functional nuclear bodies, like lamina, nucleoli, PMR body, Kaha body, speckles, etc. But how these large-scale chromatins are interacting and with these nuclear bodies and their proximity to these subnuclear structures remain quite poorly annotated. But we all know that these interactions are functionally very important for the genome function and cellular identity. So there's a really a large gap between what we can infer indirectly in a way from existing genome-wide mapping methods like CHi-C versus what we can see directly, right, under the microscope using imaging in terms of the spatial position of the chromatin in interaction with subnuclear structures. So the overarching goal of our center projects is to bridge this gap to provide a more complete view of how chromosomes are spatially organized relative to multiple nuclear bodies. So this is really the goal of this of our UM1 center project. So here's an organization of our center project. These are 5 major pillars of the project and what we do, how we organize, integrate expertise from different groups working towards a common goal. So, and then there's some diagram here on the right-hand side showing different parts. So first is that we're going to generate in-depth the so-called reference maps for a selected set of systems, cell type based on detailed genomic and imaging surveys. And then these data will be coming from whole genome mapping approaches, for example, TSA-seq developed by Andy Belmont's lab, and also functional assays like DNA replication timing RepliSeq from David Gilbert. And we have various types of imaging technologies from, for example, Susan Rafalski in Allen Institute of Cell Sciences, these live cell imaging approaches from Thomas Daly at NCI, these high throughput FISH approaches, and also certainly from Ting Wu, who pioneered these oligopin and oligostome approaches to trace the chromosomes trajectories. And all these data sets will be integrated in several different ways. Frank Albert's lab from UCLA is going to build these structural models by integrating both imaging and genomic data. And my lab will develop these machine learning approaches to collect these various types of data sets and then try to establish causal connections between DNA sequence, structure, and also function. And eventually, all these data sets will be hosted in a visualization platform, what we call Nucleon Browser, which Marc briefly mentioned. And we're working closely with Jason Swetlow's lab to develop the next generation visualization platform to organize these multimodal data sets. And all these will inform our experimentation, and there's a iterative process, and then the validations, perturbations can also inform the next round of data generation and computational method development. So I think I covered all these points I listed below. So these are really the three, actually the main research aims in our project. And these are the five components that I that described to you just a minute ago. And this is just a current screenshot of the Nucleome browser or visualization platform developed by Xiaopeng Zhu and Yang Zhang in my group, and we work closely with Jason Swerdlow's lab at University of Dundee so this is really a very versatile and powerful platform that can organize various types of data. It's not just the genome browser of the genomic data, but we also connect with the Omero image viewer developed by Jason's lab, and also the 3D genome structure viewer that you can look at these different types of models, like from Frank Elver's lab. This is actually exactly from Elver's lab, these a structure model. And then these operations can be synchronized. So it's a very interactive exploration experience. And we're also working on some other components to make this even more flexible and powerful. And this hosts public data in a distributed manner that you don't have to host them in one place. And you can also host your own private data. So investigators can look at their own data set before publication together with a lot of other public resources in the same platform. So this is the URL. If you have interest, please take a look. And this is our kickoff meeting in our center a few months ago. And here are our Twitter handles, so feel free to follow us. And then we really look forward to collaborations. We're already doing this within the 40N consortium with other investigators in the 40N, but we're also very much looking forward to interacting with others in the community, especially our colleagues in other countries, that to foster further collaborations. And our projects can provide, I think, mostly three different techniques. One is certainly the type of data, both genomic and imaging. These are experimental technologies. And second is computational expertise from machine learning methods and structure modeling approaches. And finally, the visualization platform that I briefly mentioned to you. So in the next maybe 15 minutes or so, I will very quickly go through my own research, and I'll highlight two work that we recently published. One is in press right now. It's going to come out in maybe a couple weeks. So my research, the research in my lab, as Marc mentioned, is primarily focused on developing machine learning computational methods in general to study genome structure and function. In recent years, we have particular interest in the spatial compartmentalization of the chromosomes relative to multiple nuclear bodies, and also how the 3D genome and these organization and also their variation in different cellular conditions and at single cell resolution. And we're also interested in the interaction between chromatin and other biomolecules like proteins and RNA, how these structures and organizations have evolved in mammals, but I will not talk about the last two parts in today's presentation. I'll highlight two recent work. One is called SPIN, recently published in Genome Biology, and the other is a method called Higashi for single cell 3D genome analysis. This is coming out in Nature Biotechnology. So as I mentioned, from CHi-C analysis, by looking at the PC1, eigenvectors, you'll be able to derive A and B compartments. And this can actually also be refined, from Erez Lieberman-Eden's work in this Rao et al. Cell 2014 paper, by using a very high coverage CHi-C in GM12878. By looking at the interchromosomal contact maps, you'll be able to subdivide AB compartments into these so-called subcompartments, right? There are 5 major subcompartments. B4 is mostly it's a small subcompartment, chromosome 19, but across the genome, there are 5 major subcompartments. And this method needs very high coverage. And later on, this graduate student Kyle in my group developed a machine learning method that you can actually enhance the signals for low coverage and modest coverage CHi-C data sets that you can also call subcompartments. But this is still not ideal, or something that there's a major gap between what these CHi-C compartments are offering and what we really like to know about the interaction between chromatin and other subnuclear structures, because Hi-C subcompartments do not explicitly tell us the compartmentalization patterns relative to multiple subnuclear structures. So this work is led by a graduate student graduating soon, Yuchuan Wang. So I'll very briefly go through this. And this is actually a outcome from our work in the phase 1 for the nucleome by collaborating closely, working with 3 other groups, Andy Belmont's lab at University of Illinois, Boston Steen's lab at Netherlands Cancer Institute, and David Gilbert's lab at the time at Florida State, and his lab is now at San Diego Biomedical Research Institute. So we utilize 2 types of technology. One is called TSA-seq that uses antibody staining combined with TSA reaction timer signal amplification to generate these diffusible biotin labeling near the target of interest, so that the degree of biotinylation will become a function of distance from the target. So by generating these kind of genomic maps, you see that there are these signals. If the signal is high, the genomic signal is high, it means that this particular genomic region is closer to the protein of interest. In this case, this is the SUM protein, which is localized in nuclear speckle. So in this region, I think about 4 million base pair long, you can see that these 5 different probes are reflecting the trajectory from the nuclear periphery all the way to nuclear speckle. There's another technology, older technology developed by Osman Steenzel's DAM-ID, which is based on the creation of a fusion protein consisting of DAM and a chromatin protein of interest. So, for example, if you use lamin B, and then you will be able, by sequencing, you will be generate these kind of maps to measure the contact frequency between chromatin and a chromatin protein of interest, because the DAM methylates the A in the sequence GATC. So the methylation sensitive restriction enzyme will be able to detect them, and then through these kind of sequencing, you'll be able to generate these genomic map as well. So you can define LADs, etc., but we use these input signal and integrate it with a TSA-seq signal for our computation. So the goal is that we try to generate a genome-wide segmentation. The goal is actually very straightforward: genome-wide segmentation into these color bars. So each color bar is referring to a distinct spatial position in the nucleus relative to multiple nuclear bodies. So in this case, the input includes DAM-ID, lamin B, DAM-ID nucleolus, TSA lamin B, and TSA-seq SUM protein, and what we call the spin state spatial position inference for the nuclear genome. So here's how it works. The input includes the genomic signals, including TSA-seq and DAM-ID. We also involve Hi-C data. The intuition is that if 2 genomic loci are spatially closer to each other, sharing nuclear compartmentalization states, they will have higher Hi-C chromatin interaction contacts. So we build this graphical model, right? It's a Markov random field. Each node represents genomic bin, and each node has a hidden states. These hidden states are referring to their position relative to multiple nuclear bodies in the nucleus. And we have observed the signals, and these observed signals are the signals from TSA-seq and DAM-ID. These edges are either the 2 genomic bins are right next to each other on the genome, or they have high inferred Hi-C interaction, these dashed red lines. So we use this formulation. Nodes are genomic bins. The edges represent Hi-C interactions or adjacent chromosomes. And we use this Markov random field, right? So this is probabilistic graphical model basics. So we assume that what we want to do is to maximize the joint probability based on observed data. O here is the observed data, and H is the hidden state. So there's node potentials and edge potentials. The parameters can be estimated through expectation maximization. And eventually the hidden states can be inferred using loopy belief propagation algorithm. So here's the resulting P562. These are the states and their corresponding, and we identified 10 major states from the 3 major nuclear bodies, and also their genome-wide distributions. And the colors here are showing their perceive the positions in the nucleus relative to nucleus speckle, nucleolamina, and nucleolus. And the states, you can see that we also confirm by comparing to some imaging data from Andy Belmont's lab, and you can see that this laminar state, speckle state close to the sun protein. And we also have these interior active. These are the states that are in the nuclear interior, but it's not like touching the nucleus speckles. You can see that it's not. It's away from the sun proteins. And one of the advantages of these spin states is they can stratify various types of functional and nuclear structure data. For instance, they stratify histomarks, various types of histomarks. Each row is spin states, and each column represents histomarks. And also, it provides spatial interpretation for CHi-C subcompartments, as we show here. And it also nicely stratify the multi-fraction replication timing. So the DNA that's being replicated early in the S phase is more towards the speckles, and late ones are more towards the lamin-like and lamina states. Our analysis in the interaction with nuclear bodies highlights the importance to have an integrative lens to look at high-order genome structure and function. So in a work last year from my group, we developed this approach to integrate 3D genome organization with transcription regulatory networks. So you can look at some of the network modules by giving them the context of spatial position in the nucleus. And they form these spatial domains or modules that are also functionally, they also have functional implications. And so so in a way, it highlights the advantage of looking at 3D genome together with data from measuring genome function like transcription and replication. So in the preview article we wrote about 2 years ago, when the ERCE paper developed published by my colleague Dave Gilbert's lab in our project, we wrote a preview article to highlight the importance to look at genome structure and function in a more integrative manner, because a lot of these formation mechanisms for the 3D genome, the cis and trans factors that are playing an important role are not quite clear. So it is important to make sure that these different factors are being considered in a more coherent and integrative manner. So in the last couple minutes, I'll highlight this work led by a graduate student rotating my group. So all of the results I present so far, these are in bulk estimate. We all know that they may obscure single cell level features, and especially there's no direct measurement of the variability among a cell population of various types of 3D genome features that we would like to see. But the 3D genome, single cell 3D genome data is extremely challenging to analyze. So we developed this machine learning approach to tackle this. So here are some challenges for analyzing single cell CHi-C data. The variability of the single cell. We wanted to review the variability of the single cell, the multi-scale feature of the 3D genome, right, from loops, tags, compartments, etc. And we also wanted to integrate it with other data types, not just the 3D genome aspect, but genome function like transcription or even other epigenomic measurements into the same framework. But the challenges involve high dimensionality of the data, dependencies of the entry in this content map, and the data is extremely noisy and sparse at the moment because of the limitation of the technology. So here's an example is the bulk CHi-C content map. You can see some structures from the content map on the diagonal and even off diagonal ones. But if you look at a single cell CHi-C content map, it's very sparse. So our goal is to enhance this, right? So the typical single cell CHi-C analysis framework involves generating embeddings of the cell. So you can study, say, from the cell population, are there intrinsic clusters of the cell into cell states or cell types? And we also want to impute these sparse content maps so that you can derive more refined 3D genome features at the single cell resolution, so you can look at their variabilities. And our solution is so-called hypergraph. So we all know graph. In graph you have nodes and edges, but in hypergraph you also have nodes, but the edges are hyper edges in the sense that involve multiple nodes. Here's an example. This is authorship example. These nodes are individuals, are scientists, and these yellow circles, these are edges. These are hyper edges. So one hyper edge may involve multiple nodes. So one paper may involve multiple authors. So each one of these yellow circle hyper edge represents co-authorship relationship. You can also have events, right, to using hyperedge to model events, human location activity. So analogy I often give is that if I give you some profiles or information of individuals and their online activity, like what are the posts they like, or the tweets that they're retweeting, they like, can you predict if these individuals on the social network is going to form a group chat, right? So a group chat is like you can represent as a hyperedge. And that's exactly what we want to do for single cell CHi-C analysis. So this is so-called hypergraph representation learning. So our aim is to learn the embedding of the nodes in the hypergraph and also predict the existence of hyperedges given the node embeddings. This is similar to the analogy I give you about the individual activity on the social network, and then to predict if they're going to form a group chat. And this work was, the model itself, generic machine learning model, was published earlier in the machine learning conference ICRR. So in this Nature Biotechnology paper that's coming out very soon, we consider this triplet as a hyperedge. So here a triplet involves 2 genomic bin and 1 cell node. So we want to do is, and you for a collection of single cell contact maps from single cell CHi-C, you create a whole hypergraph. And in this hypergraph, these hyper edges are all triplets, 1 cell node, 2 genomic node. This in each hyper edge indicates these 2 genomic nodes, I and K, for instance, they are interacting in the cell I, right? So as I mentioned before, the sparsity is high, and then a lot of these hyper edges are not there. But our goal is to predict if a hyper edge should exist, is there. So essentially, you want to predict if the hyper edge, what is the probability of a hyper edge given the information of these individual nodes. This is similar to the examples I gave you before. So once we have this, we can also generate the embedding of the cell, and you can produce these cell type embeddings. You can analyze complex tissues based upon their 3D genome features. And from imputation, you can derive features like compartments and pads. You can look at their cell-to-cell variability. In addition, you can compare with other omic modalities into the same analysis framework. So the hypergraph representation learning I mentioned to you before, the 2 questions, now they are turning to the 2 problems you want to solve, the embedding of the cell and the imputation of the contact map. Here's some examples. This is data from also a 40 nucleome investigator team in Bing Ren, who leads another UM1 center project in phase 2, 40N. So the first line is the original single cell CHi-C contact map, and the second line is the imputation result from Higashi from our approaches. You can see that we can drastically enhance the signals, and you can derive a lot of useful information from after the imputation. And on the left-hand side, this is showing the when we call the installation scores in individual cells. Each line is an individual cell, and each column here is a genomic loci. And after the imputation, you can derive different kinds of variability of the TET-like domain boundaries. And the yellow box here is showing a type of domain boundary that present in some cells but absent in some of the other cells. But this red box here highlights the variability of the domain boundary, where it gradually shifts along the genome. So these are 2 types of variabilities of the TET-like domain boundaries in this cell population WTC11 iPS cell. But if you don't do the imputation, you just look at infer the installation score from the original sparse contact map, you can see that the signals are very, very noisy, that it's very difficult to derive useful information. So we also apply this approach to a real data set in, this is in human prefrontal cortex, generated from a previous publication from Joe Ecker's lab. And in their work, they stated that if you only use single cell Hi-C, you're not going to be able to group, cluster these cell types nicely. But by using our more advanced Higashi approach, we found that actually after imputation and embedding, you'll be able to nicely separate these subtypes, especially neuron subtypes, the inhibitory and accessory neuron subtypes that were previous method were not able to do. So this is just an example of a GAD1, GAD1. This is a marker gene for inhibitory neurons in SST, and you can see that. And then there's a domain boundary. So the red color means the insertion score is lower, indicating that stronger domain boundary is specific to inhibitory neurons. And if we look at the contact maps, you can see that the strength of the boundaries is stronger in inhibitory neurons compared to accessory neurons, for instance. So this is the summary of the Higashi. So it's a new machine learning approach by also incorporating latent correlation among different cells for data imputation. And this is a more advanced approach to delineate cell-to-cell variability from single cell Hi-C data. And I didn't elaborate too much, but it does have the advantage for incorporating signals jointly profiled in the same cell, for instance, methylation from these human prefrontal cortex data I presented. And overall, this framework provides a new analysis approach to derive single cell level structure function connections. We have a visualization tool that to help other people, when you try to debug your code or look at these individual contact maps together with other features, you can use this tool to do that. So I'm going to stop here and leave some time for questions. And I think I mentioned the names of the group members in my lab who developed the work that I mentioned. But none of this will be possible without working with many other labs that we interact with within the 40N and in large community. In particular, in our UM1 phase 2 project in NIH 40 Nucleome, these are the labs that I mentioned, and also other investigators in the 40N network and beyond in the larger 3D genome community. So thank you very much. I'll be happy to take questions.
So thank you very much for such a brilliant talk. The first question comes from Ilya Flammer. He's asking, how do you know that you're not just making up some data for each cell and these contacts that you infer were likely really there, but just were not captured?
Great question. So I didn't have time to go through all the results, but we did have quite detailed evaluation in the publication. One thing that we did, I think it's more realistic, and I really like that part of the evaluations, that we use the imaging data as part of the evaluation. So what we did is that these multiplex 3D imaging data from, I think we used the two data sets from Xiaowei Zhang's lab. One is Bingtu et al. And the other Su et al. So you can take the 3D imaging data as ground truth, right? So that's the ground truth that we wanted to recover. So what we did is we downsample that. We also only sample a small fraction of those contacts measured by imaging. And then we feed that into the Higashi, actually some of the other competing methods, to see their imputation performance. As you said, what are the false positives and false negatives that you expect to see from these methods? So that's a way to evaluate the approaches. And you can certainly downsample that in different levels. If you downsample to very, very sparse contact, and you expect that the false positives may increase, and that's what we see. However, I think our approach still outperforms other existing approaches for that. So to reiterate, so we do have an approach to control that, and we have a sense how well it does, because I think using imaging is a good way to evaluate these imputation methods for single cell 3D contact maps.
Yeah, really nice work. Beautiful. I had a question because you didn't touch yet on combining it with gene expression, single cell and heterogeneity, such as transcription bursting. So are you considering to include this? We are very interested in this, and yeah, working on the genome-wide transcription bursting. So how is the status there?
Okay, so let me try to answer that. So I think first of all, this framework does have the flexibility. If the measurement is jointly profiled with single cell 3 genome, this framework is readily, it can incorporate those information into the hypergraph framework that I mentioned. In our current work, we did perform some analysis to correlate the variability of the 3 genome structure with the variability of expression in several different cell types. So one of us, but of course those are more global correlations. For instance, the level of variability of cat-like domain boundaries and AB compartmentalization versus the variability of gene expression. And because one data set we use is IPSL, it can involve in differentiation. So we also look at the type of genes that are showing higher variability in WTC11 in IPSL and their role in differentiation. So there's some analysis on that by correlating the cell-to-cell variability of 3D genome features versus the cell-to-cell variability of gene expression from single-cell RNA-seq.
Okay, thank you.
Beautiful. I have actually two questions, if I might. The first one is scientific, going back to your Higashi approach. I wonder if you have tried to look at the data from Peter and Amos, Fraser and Tanai, on single cell data, where they look carefully and they found the cell cycle very beautifully. Your Higashi approach should also find it. Have you tried? When you do the embedding, if you find the same embedding that they find without actually enhancing the data?
Yes. So I didn't present that part, but we did look at one cell cycle data. I don't think that's the one from that particular paper. Yeah, but we do see that if the single cell is you profile a cell cycle, you see the embedding will manifest the sort of the cell cycle there. There's a gradual transition of the cells in the embedding. Yes, but yes. So, but I think again, so this can be applied to some of the other context where you expect that there is some gradual transition of. It's like these from single cell RNA-seq, these pseudo time that you can also use 3D genome to reflect the cellular transitions. Yes, to answer your question.
What Amos did. Beautiful. Looking forward for the paper. The other question is more technical and probably pertinent to many of us. So you guys at the 4DN in NIH are way advanced than many others in terms of data integration and data standards and data sharing. So have you sorted out how to help mostly experimentalists, not necessarily bioinformaticians, mostly experimentalists to annotate correctly their data sets and to upload them correctly? So do you have automatic ways of doing this? Do you guys have some rules that we could all follow? What are your experience of that, Jian Ma?
That's a tough question. I don't think we have, personally, we have a solution yet. And this is also a question for the whole consortium and community. Yeah. I know there's a lot of work from the what we call DCIC, the Data Coordination Center, led by Peter Park at Harvard Medical School. So their center, the DCIC Data Coordination Center, is working with the community to define a lot of these data standards, metadata annotation standards for data in multiple modalities, basically in genomic data. Even for genomic data, once there's a new assay, what are the information that you need to collect in order for other people to analyze or reproduce, or for integral analysis? And also, I think it's ongoing is the imaging data, like what type of metadata you need to provide for the imaging data, for storage, for sharing, for dissemination, and perhaps more importantly for integrative analysis with imaging data, because that's really one of the main expectations and goals for the NIH 4DN, is to integrate the imaging and omic data to have a more complete view of the 3D genome. And we're working on some part of that, and my group and also others in our Center for Jason Swallow's lab is taking a lot of leadership roles in defining these imaging data standards together with other groups, like including Ting's lab. And for the genomic side, I think we're also working on certain aspects, and some of them are coming from these working groups I mentioned before. For instance, from the predictive modeling group, we are also interested in knowing what type of information we need, right, from the data set, what type of annotations you need in order to have a more comprehensive, unbiased view, and to also to enhance interpretability from these machine learning models. So I don't know if that answers the question, but it's sort of all over the place. That I think these are also the ones that we're considering. Yes.